there is something called the hallmarks of aging. It's 16 different processes, like the breakdown of protein. Like we don't fold our protein as well. There's damage to the DNA itself. There's the epigenetic changes that I've been talking about. There's changes to cells, senescent cells, in the immune system. There's a whole host of predictable things that happen. I would say that there's new science coming out that suggests that of all of these processes happening, that epigenetics might be the root cause, which I think kind of answers your question. What happens in the epigenome, these changes to DNA methylation, may actually be sort of the root cause drivers of all of these downstream phenomena that we're seeing that we refer to as the hallmark of aging. So
Welcome to the show.
I am so thrilled to be here. Thank you, thank you, thank you for inviting me, and I'm looking forward to our conversation.
You start off the book with a pretty simple statement. You say, you can become younger. And I guess for most people that hear that, they think, I mean, it's possible in the film, The Curious Case of Benjamin Button, but how can people become younger? Like to some people, that might sound crazy. How is it possible?
Yeah, I know. I know. Well, we actually see it all the time. This is funny. I haven't actually ever expressed this thought in an interview, but it's just jumping into top of mind. I mean, we see our friends and family get inspired. They take up a health kick, they start eating better, and, maybe they see a doctor who practices integrative medicine and they just start looking better. I would argue that they are probably reversing. They're probably getting in there and making some changes. So I think that we are engaging in this all of the time in our lives. Those of us who choose to take it on, and the exact opposite happens too, right? We see friends, maybe, if you go back to a school reunion, you see friends that you can't even recognize because they look so much older. But then you see other friends who basically look the same. We actually have it's been studied. Our ability to recognize the aging process has been studied. So facial recognition, uh, identifying age through facial recognition has some scientific evidence behind it. So when you look at this person who you went to school with and they look so much older, in fact, you're actually probably right, as amazing as that is. But what's happening now in science, and it's so cool and extraordinary, is that we're able to reliably measure the rate of aging, how fast we're physically aging. And this is called biological aging. It's different than chronological aging. We can't do anything about, how many birthdays we've celebrated. Try as we might. My sister always says she's 29 again. We can't do anything about that. But the rate at which we're aging, it turns out that we can actually influence it. And of course, our study got a lot of attention here and across the pond over in the UK. I was interviewed quite a bit when our paper came out, because we were the first to actually show that a diet and lifestyle program could slow down biological aging in our study participants as compared to control.
There's so much that I want to kind of pick up on from there. But what you said is really interesting because I guess what you're trying to imply, if I've got this right, and please feel free to correct me,
Mhm.
is that essentially how young we look on the outside could be a predictor of actually what our biological age is on the inside. So if you have a friend,
Yeah.
as you said, that looks 10 years older than what they are, that could be a sign that perhaps they're not aging biologically as well as perhaps they should be. Have I got that
Yes.
right?
Yeah, you got that exactly right. I mean, it's really extraordinary. And now we're able to measure it. I want to just point out one other thing too. Our chronological age. So now that we're in the era of being able to measure biological age and we're able to look at interventions that can slow it down and turn it around, chronological age is going to lose importance. We're not going to care. When we go to our annual physical with our physician, it's not going to be the chronological age that we care about or that we're talking about with our friends, et cetera. It's our biological age that's really going to take front and center stage.
Right. So you go to a doctor's office, they'll do a series of biological tests, they'll test your age, and then that will give you a different age
Yeah.
compared to, for instance, how many birthdays it's been since you first had your birth
Yeah.
certificate.
Yes, yes, yes. I mean, there are biological age clocks are just accelerating. I mean, it's extraordinary. Even from our study, which was published in 2021, there's second and even third generation clocks now. There's a clock called the Grimage that is aptly titled, "It predicts time till death." So yeah, you want that as young as possible. But indeed, I think that you're correct. We're going to be getting those numbers from our doctors, and eventually I think we'll be able to access them ourselves. I mean, ideally in some future time, this is my Aura ring, I'll be getting my BioAge data right here. And I'll be able to tell, what activities, events, my sleep, et cetera, all of the input that goes into this now, is it, pushing biological age fast, or is it slowing it down?
Pretty remarkable. I mean, the fact, the idea that there's a test called a Grimage that can tell, I mean, essentially, probably, I imagine, with some predictive accuracy, how long you
Yes.
have until your death. That
Yeah.
sounds pretty remarkable.
Or time till illness. There's
Yeah.
another clock called PhenoAge, which looks at morbidity and mortality as well, but how your good years left. Yeah, it's
I,
getting very,
yeah.
and there's even a rate of aging clock that I actually like quite a bit, called the pace of aging, which, if you're greater than 1, you're aging faster than chronological age. So we all want to be less than 1. And this one is very responsive to different interventions as well. And so it's something that we could test repeatedly, and make sure we're living our best lives. Unfortunately, it's kind of still cost prohibitive
Mm.
to access these clocks, but I'll say, just from the time in our study, the clock that we used, or the test that we used, was in the research setting only. No, that's no longer the case. And back then in 2018, 2019, it was $1,200 per. It was very expensive. They're down to maybe $200 now. So expect these numbers to continue to drop, and for them to become more and more available to us.
I love it. Before I jump into your study, I wonder if you could just explain, perhaps, to the people listening, someone is listening to this conversation, they going, wow, I mean, how can there be a test that tells me what my biological age is? How can there be a test that tells me, what my rate of aging is, as
Yeah.
opposed to, just birthday to birthday? And even the craziest one, how can there be a test called the Grim Age that tells me, perhaps, how long it is until I get seriously sick or even die? Could you
I
explain, like,
know.
kind of the biology in that?
Sure. Yeah, that's such a good question. It's a great question. We're looking at gene expression. This is the era of looking at what genes are on and what genes are off. It's called epigenetics, epi above, genetics, your genes. So uh, patterns of gene expression, and specifically, so when we're in epigenetics, there are a number of variables that regulate gene expression. Epigenetic marks, they're called, a number of different biochemical marks that influence what genes are on and which genes are off. But the most studied and one of the most resilient and arguably the most important ways that we turn genes on and off is something called DNA methylation. And the clocks are based on different patterns of DNA methylation. Um, the original clocks, the earliest clocks, were trained against chronological age. And so basically you can just think about a massive, massive data set of DNA methylation marks of thousands and thousands of people were, just fed into this big computer, with the question being, what pattern of DNA methylation marks correlates most strongly with chronological age? So these were the first clocks, and actually the clock that we used, the Horvath multi-tissue clock, which was the only clock available in 2018, which was like yesterday. It's kind of crazy how fast this technology is
Right,
is
right.
moving. Um, so Steve Horvath, who developed this clock out of UCLA, University of California in LA, asked, basically, what pattern of DNA methylation marks can most closely predict chronological age, and he was able to do it with almost complete accuracy, 0.96. So very very tight.
Oh,
So this is that they looked in utero, so they looked at fetal DNA all the way to, centenarians, and they were able to predict across the timeline different changes. And there were just 353 methylation marks. They're referred to as CPG sites because on the DNA it's on a cytosine when the cytosine is next to a guanine and it's connected by a phosphate. That's the piece. So CPG, they refer to them as CPGs. So in the Horvath, the first Horvath clock, which is what we used, there are 353 sites, and it's very predictive, very close to chronological age, except it's actually not perfect. There's that little bit of wiggle room, right? That 0.4 because it's 0.96. And what they demonstrated is that these DNA methylation patterns, even in this first clock, were more predictive of morbidity and mortality than chronological age. So even at the very first iteration of the clocks, they were better predictors of health outcome and lifespan. Isn't that amazing? And so they've refined the clocks over time, and they've been able to look more closely at, like, the Grimage is time to death, the PhenoAge looks at morbidity carefully, or the pace of aging. It's called the Dunedin Pace of Aging clock out of Duke University, and they've been tracking a cohort of individuals from the time they were 3 and then now in their 50s, and they've established methylation patterns to look at the pace of aging in this population. So they're just getting more and more refined. It's all rather extraordinary. But let me stop there and see if you have any questions on that, if that's relatively clear.
well, imagine a question some people would be wondering is, have you done a Grimage test?
um, I have, yeah, that's pretty funny. But what's interesting? I can't off the top of my head. I can't even tell you. It hasn't. I've gotten a lot of clocks, so I can't remember. I can't actually tell you um, what my age was. That's so funny. The Horvath clock, kind of the flagship clock. I've consistently been younger, like pretty
Nice.
significantly younger than my biological age. And I recently just did another clock and similar, consistent. So I've put a lot of effort into kind of living what I'm espousing in my book. And, so far so good with my biological age. But yeah, I can't, I'll have to look up my last, when I got the Grim age. I have a ton of data. I have a lot of data.
Amazing. Well, to be fair, I mean, I guess if you're not too worried about what the results that it gave you, I mean, it can't have been that bad. So you must be scoring pretty
Right.
well. Um,
Yeah.
but I'd love to kind of pick up on some things with it, because it seems to me like going through your book and exactly what you just said, two things are really key. And I guess before we jump into your study, you mentioned epigenetics, above the gene, and DNA methylation. These seem really key in the process of aging. Can you explain why?
Yeah. So it's very, very interesting. So again, going back to what Horvath first discovered, he just published this clock in 2013. This is new, but he came up with these 353 DNA methylation sites that were a more rigorous predictor of morbidity and mortality than age itself. So that was his first clock. He's gone on to develop other clocks. He actually has a mammalian clock out now. So across all mammals, there's a predictive collection of methylation sites that correlate with biological age and aging. It's extraordinary.
He's being
Um,
productive. He's a
yeah,
productive man, Steve.
yeah. He's working with a lot of really brilliant scientists, but no doubt, yeah, it's really interesting the work that they're doing. They're developing clocks for all sorts of
Mind-blowing stuff.
species.
Yeah.
So the question is this clock, are these clocks, these new technologies that are available, these new assays that are available, are they surrogate markers of aging? Are they just sort of suggesting an aging phenomena happening elsewhere? Are they just measuring it? Or are they actually the cause of aging, the drivers of aging? And that's your question to me.
Mm.
What are we just sort of getting a shadow of the aging process happening somewhere else, or are these guys really in the driver's seat? And there is something called the hallmarks of aging. It's 16 different processes, like the breakdown of protein, like we don't fold our protein as well. There's damage to the DNA itself. There's the epigenetic changes that I've been talking about. There's, changes to cells, senescent cells, in the immune system. There's a whole host of predictable things that happen. I would say that there's new science coming out that suggests that of all of these processes happening, that epigenetics might be the root cause, which I think kind of answers your question. What happens in the epigenome, these changes to DNA methylation, may actually be sort of the root cause drivers of all of these downstream phenomena that we're seeing, that we refer to as the hallmark of aging. So it may be that, yeah, we want to be really thinking about gene expression and optimizing it. And that we might be really going towards root cause, which is something obviously in functional medicine we're extremely enamored of. And I'll say one other interesting piece, and that's, there's a handful of studies. Actually, there was one out of University of Cambridge, and I think an associated lab, looking at skin age. So what they did was they took 50-something-year-old skin cells, and they used something called Yamanaka factors to influence DNA methylation and gene expression. And they reversed, this was in vitro, this was like in a petri dish, these skin cells. They were able to reverse the age of these skin cells by, I think 30 years, maybe more than
Wow.
30 years. Yeah, so in a petri dish, they turned back time by changing DNA methylation and epigenetic expression. This has also been shown in mice. There was an age-related optic neuropathy that was reversed in mice. So they were blind because they had this age-related optic neuropathy. Again, they were given these compounds called Yamanaka factors that changed gene expression, changed DNA methylation and demethylation, and they reversed the optic neurons. They made them younger, and they restored vision in the process.
And perhaps this would be a great point to jump into your study. So this obviously was one of the foundational aspects that led to this book. So when I look into your study, I mean, it's pretty crazy that you took a group of people, and what you found was that in just 8 weeks, you reduced their biological age by 3 years.
Yeah,
which is pretty bonkers. So could you talk to us about this?
Yes, yeah. So we had designed, years prior. In fact, I actually taught on this in Ireland. There was a functional medicine conference back in 2016. I brought this over there, and I taught in other parts of the world. Like, people in functional medicine, clinicians practicing functional medicine, were interested in this. So years prior to conducting our study, I was reading the literature on epigenetics and particularly cancer. At that time, there was no evidence that we could change biological age in around 2013, 14, 15, 16. The first study published wasn't until 2019. And then ours came out a couple of years after. But it seemed to me in functional medicine that we had a responsibility to thinking about epigenetics, to thinking about optimizing gene expression. Like, we really had a responsibility to look at what interventions we were doing, what supplements we were prescribing, our diet and lifestyle, to make sure that we were doing this as healthy as possible. And at that time, it was just theoretical, but it was so important to me in my clinic that I worked with our nutrition director at the time, Romilly Hodges, on designing what became the methylation diet and lifestyle, which is now the Younger You program. And everything, so we just scoured the literature for evidence on nutrients that influence DNA methylation and diet and lifestyle factors. So that's how it started. And then in late 2017, I was given an unrestricted grant to study it. So at the time when we developed the program, of course, I wanted to look and see, are we changing gene expression? But there were no tests available. We couldn't send somebody to the local lab. Like, we couldn't do anything in-house, but we believed in what we were doing and the intelligence with which we designed it. And then I was given an unrestricted grant in late 2017 to study it. And so we were able to hire a clinical research center to, enact this randomized control trial. It was during our study. I mean, so we knew we could measure biological age, but we didn't anticipate that we would have reversed it. Because thus far there was nothing in humans to suggest this was possible. So it was like mid-study that the first paper was published. on this being a possibility, and it was, fanfare the scientific community over. I think they were written up in Nature. Their first the study was published in Aging Cell, if I recall correctly, which is a very top tier journal in biogerontology. And we knew we were going to look at these clocks, but I guess we started to have some idea that maybe we could make a difference. And so one of our first points of analysis was in looking at this original Horvath clock, and we were dumbstruck when we saw the influence that we were able to have on slowing it.
And again, just to kind of underscore the results, I mean, reported of 3 years of biological reduction in age in just 8 weeks. So I wonder, perhaps you could talk to us about the study design, perhaps, how the participants, the kind of changes that they went to kind of see these results, because I imagine you don't wake up 3 years younger overnight. Can you talk to us about that?
Yeah, so we wanted to look at healthy people. We really wanted to kind of zero in on the changes that happen with aging. So we collected, we had a healthy cohort. And so can we? I mean, arguably, it's a heavier lift to reverse biological age in a group that's already really healthy. We know that when you're not healthy, you're aging faster, and you have sort of a more potential for bioage reversal if you can actually eradicate whatever disease process is happening. So we were looking at healthy men. And so they already had some good habits. They already were open to eating, while they were already moving their bodies to some extent. So because they were healthy guys, but this was a big change. So to influence DNA methylation, the diet program needs to be packed with certain nutrients. We want every forkful to be full of epi-nutrients. And I suspect you're going to want to ask me more questions about exactly what the diet entailed. And I'm happy to talk to you. But I wanted every single forkful that these guys consumed to be like massaging their gene expression, to be massaging DNA methylation as best as we perceived in the literature. I mean, it's kind of a small literature at this time. We know that sufficient amounts of exercise has a powerful influence on DNA methylation. We know that sleeping, if you're sleeping well, you are supporting ideal gene expression and DNA methylation. If you're not, you're damaging it. Stress is a huge driver of negative changes to gene expression and DNA methylation in particular. So we prescribed a meditation program. People who meditate, people who engage in some intentional stress reduction habit, be it meditation, Tai Chi, yoga, even an exercise, et cetera, these people are biologically younger. So we just built brick by brick a program that encompassed those things that we were reading in the literature that could make a difference. Plus, we also needed it to be adoptable. We needed it to be doable. Certainly scientists studying this would say 8 weeks is ridiculous, Kara. Like, you can't change DNA expression in such a short period of time. But my read on the literature was that we could. There was some suggestion that we could, and I can give you those examples if you want to. I mean, we can change gene expression relatively quickly. But the other piece was, as a clinician, somebody working with humans every day, 8 weeks is about the longest time you're going to get somebody to adhere really pretty impeccably to a fairly rigorous intervention. So if we wanted to push them 6 months out, it's, that's tricky. We would get people falling off and perhaps not reporting accurately. And so we did that. And we used coaches, we used nutritionists from my practice to meet with our study participants weekly or more if they required it, just to make sure that they were adhering to the program.
I love the term epi-nutrients, and I also know from going through your book that you're a big advocate of liver, which I love, by the way.
I'm kind of reluctantly. I mean, I don't love eating liver, but like it's brilliant. I mean, it is a multivitamin in a food matrix. I mean, it's the most nutrient dense food in the world, really. I mean, there's nothing that compares to it. So we couldn't we couldn't ignore it, even though I'm not going to be cooking myself any liver and onions anytime soon. The fact is, it's brilliant. I like a good pate.
I agree, and I'm a bit of a contrarian with this because I actually love the taste of liver. I'm such a massive fan. It's one of the reasons I could never be vegan. But there you go.
Good
But
for you. Yeah, it's smart. It's you know, we were before we were able to synthesize vitamins, people got injections of liver. I mean, that's how we treated B12 deficiency or pernicious anemia or any of these micronutrient deficiencies. I think you just got a big shot of liver into your butt. I
wow.
mean, it's really amazing, like emulsified liver.
So is it the case that we want to, in essence, get as much of these vitamins and nutrients as possible without, I guess, the excess of, excess calories, excess sugar, excess body fat? Is that
Yes.
the aim for us?
Yeah, that's right. That's a really important question. So what we're removing, what we're taking out is really as important as what we're adding in. So yeah, 100%. Yes. If you've got poor blood sugar control, if you're carbohydrate intolerant, your blood sugar, your insulin, et cetera, your A1C are elevated, that is a pro-aging phenomena. That's damaging to epigenetic expression, to DNA methylation, for sure. It's very pro-aging. So we need to dial that in. If inflammatory, if you're particularly inflamed or if you've got marked dysbiosis, like all of those things are impactful on longevity, on biological age, on epigenetic processes. So yes, the diet pulls out pro-inflammatory foods, high sugar foods. It's higher in good fats. There's a gentle intermittent fasting structure. So we wanted people to get into a little bit of ketosis. We didn't want it to be a ketogenic diet in the classic sense, but we wanted it to be keto leaning, as we call it, because those ketones are signal molecules for the epigenome and they're potently anti-inflammatory. We know they're, it's just smart compounds to produce.
Wow. And just kind of sticking with diet, you kind of mentioned epinutrients and you go into so much detail in all this in the book. But I wonder if you could just perhaps give us, because you have quite an interesting take on diet, I would say. I wonder if you could kind of talk to us about why you feel that in terms of this kind of Younger You program, kind of how you approach the topic of diet advice for people.
That's a good question. I think it's relatively standard in functional medicine. We want to be avoiding processed foods. You don't want to be going crazy on simple carbohydrates. What we call the Younger You intensive that's in the book is that's what we did in our study. And that's a more aggressive program than what we call the Younger You every day. So there's no alcohol in the Younger You intensive, but it's only 8 weeks. I don't want to freak anybody out. If you like to drink, if you can hold on for 8 weeks, you can, go back to having your cocktail or your glass of wine or whatever. But there's no grain or there are no legumes in the 8-week intensive. Because they can be pro-inflammatory or some individuals that they might spike blood sugar. So we just wanted to be really strict I think with an eating pattern that's understood to be healthy for that 8 weeks time. And then we loosen up. I'm a fan of legumes, by the way, for any vegan out there or anybody who pays attention to, what centenarian diets are comprised of. Legumes always make a percentage of those. So I am a fan of those, but it's just during that 8-week intensive, I want to be extra mindful for people in our world. We're not in blue zones by and large, in the Western world. But then, so layered on to those, I think standard good dietary practices are, just loads of these epinutrients. And I think that's the unique attribute. When you see what epinutrients are, and we'll talk about some of the specifics, you'll know that, these are nutrients you already recognize as being smart. There's many of them though. So I built out a nutrient appendix in the book, and there's about 30 pages of these nutrients that have some evidence for favorably regulating gene expression and specifically DNA methylation. So certainly unique to our program is, every fourth fall should be packed with these epinutrients.
Could we go into the specifics?
Yeah, we sure can. We have a dynamic dozen. So we've got sort of the 12 that we love. Let me see if I can grab one.
Please.
Here's rosemary.
Rosemary, yeah.
Rosemary, rosmarinic acid. It's not just in rosemary, it's in thyme, it's in other savory spices. Rosmarinic acid is a beautiful epinutrient. Curcumin, we know turmeric. I mean, lots of us love a good curry or a good glass of golden milk. I try to have one daily. It's a epinutrient superstar. It doesn't, well, we should talk about the difference between the methyl donor class of epinutrients and the methylation adaptogen class of nutrients. So the methylation adaptogens are epinutrients that seem to influence where DNA methylation is happening. So when you've got a lot of methyl groups on a gene, in the literature, they're always denoted as red lollipops. So if you picture a strand of DNA or a double-strand double helix, and you just see a lot of these red lollipops on a region of a gene, that's at the, and usually they're at the gene promoter region, the area that lets a gene be on or off. Lots of red lollipops like inhibit that gene from being turned on. Just structurally, you can imagine the transcription factor that would initiate transcription of that particular gene is inhibited from landing on it. So lots of methylation on a gene will shut it off. Few methyl groups on a gene or none allow that gene to be turned on. We want the right genes turned off, the genes that promote disease, we want those inhibited, and we want the good genes, anti-inflammatory genes, healthy genes, longevity genes, we want those to be on. So we need two things. We need methyl donors. We need to make those red lollipops. We need the ingredients to make those red lollipops. And back to liver. Liver is a superstar methyl donor food. Superstar. It's loaded with the classic methyl donors, including B12 and folate and choline. Really important. And it's just got a host of other important nutrients like vitamin A and iron and zinc, etc. Greens are loaded, cruciferous veggies, seeds, nuts, mushrooms, wild mushrooms, shiitake, enoki, maitake, even button mushrooms have some of these essential nutrients, the folates, the cholines, etc., that help us make these red lollipops. So we need lots of red lollipops. As we age, we actually have less red lollipops collectively. If you measure all of the DNA methylation going on, it's called the methylome. If you measure all of that as we age, we have less. So we want to be pouring in methyl donors. We want to be eating methyl donor foods every day. But we also need to direct where it happens. Classically, as we age, we start to shut off genes that we want on. Actually, in the diseases of aging, the same thing happens. We're shutting off genes. We're taking those precious red lollipops and we're turning off genes that we want on. So our anti-inflammatory genes get inhibited, genes that protect us from diseases, classically tumor suppressor genes. These are genes that we want on and clearing us of cancer. As we age or when we get cancer, we actually inhibit these genes. So how do we direct where these red lollipops are going in? And I think that these methylation adaptogens, this other class of nutrients, helps to direct where that's happening. And those nutrients, again, are the rosmarinic acid, as I showed earlier. The curcumin is brilliant. Green tea, so curcumin and turmeric. Green tea, EGCG, the catechins in green tea. Many of the polyphenols in the colorful fruits and veg that we eat, in berries and blueberries and in blackberries and strawberries, a compound called fisetin, and really on and on. I mean, these nutrients appear to have some ability to make sure that we're turning off the bad genes and turning on the good genes. And what we saw in our study population is that we did just that. So we didn't increase overall methylation in the study population, but we rearranged where those red lollipops were being laid down to something more useful. In fact, we have a whole nother paper I have to write with somebody hopefully I'll find somebody to co-author this with me. It's like kind of a heavy lift, but exploring specifically the genes that we turned on and off in our study population and really describing beyond just the biological clocks, what we did, because we made a huge change in gene expression in our study population.
That's all pretty remarkable. And I'd love to pick up on one thing you kind
Sure.
of said. And
Yeah.
I've got this thing in my mind kind of, because you mentioned liver and how it being so nutrient dense. Now, I was told a long time ago, and this has just popped into my mind, so maybe you can fact check me, that it might be bad for pregnant women to consume liver. Now, I would love to ask you, is that
That's
true?
interesting.
And if not, kind of what principles should people take for perhaps eating when they're pregnant or postpartum?
That's a great question. Wow, that's really interesting. I have not heard that. I think liver would be super smart during pregnancy because it is so nutrient dense. And there's something about the food, it being housed in within a food matrix that I think is safer than just taking isolated vitamins. That said, if we could we could consume too many methyl donors, and that could be a problem. And here's how. And we want to be careful about that. And I actually spent quite a bit of time talking about that in the book. In fact, I created what I call the Younger You hybrid, which is the macronutrient program I would recommend for eating during pregnancy, postpartum, and preconception. So it's got there's higher protein in it. There's extra carbs for all of the energy demands. It's actually more caloric rich. But I do pay attention. So we could easily consume excess methyl donors, too much folate, too much B12, too much choline, too much betaine, if we are taking a prenatal vitamin, if we are consuming fortified foods. So is the flour, is the grain that you're using, does it have folate added to it, folic acid added to it? Do you guys have a folic acid fortification program in the UK? Is there a law that requires you to add folate to flours and so forth?
Perhaps. I'm afraid
I should
I'm
know
not too
that.
sure. I'm so
I know,
sorry. I
I
don't
know.
know. I don't know.
I should,
I'll
somebody
check.
out
We'll
there.
put a
So
comment in the description.
put
We'll
a,
check.
okay, perfect, perfect. It's, I think I actually have it in the book. I researched it, but it's not top of mind and I don't want to be incorrect. Um, you may, or certainly there's other European countries that do, but there are plenty that don't. The United States does. We know we need enough folate during pregnancy to prevent birth defects. This is essential. And so United States enacted this requirement, and we did see a significant drop in the rate of birth defects here. But now we're also seeing too much folate, too much methyl donor consumption if you're doing certain things. So you're eating these processed fortified grains. If you can't consume animal dairy, and you're drinking nut milks or grain-based milks. In the US, they're always fortified with folate, with B12, with other nutrients. You're eating almost a multivitamin or drinking in a glass of nut milk. So imagine if you're eating, processed grains, which in the US are fortified. You're drinking nut milks because you can't handle dairy. You're taking a prenatal vitamin, you're overwhelming your body with these methyl donors. And so we do need to be sophisticated. We do need to be aware of it. And I'll throw out one other really interesting thing. Genistein in soy is a epinutrient of extraordinary proportions. In the intensive, we don't include soy, but if you can tolerate soy, once you go into the everyday and you can get clean, organic soy, it can be a really important nutrient. And by the way, one of the problems with liver is you need to get clean source liver. So I would say that is something that you have to look for. Can you get a nice clean, pasture raised, minimal pesticide exposure liver? So then you're eating the best quality stuff. That's important. But genistein, there's research looking, and this is in animals, so this is in mice, but genistein in pregnant mice prompted a gene expression change, an inhibition of a hypermethylation, so lots of red lollipops on a particular gene. Genistein from soy, which is not a methyl donor, acted like a methyl donor in these pregnant mice. It's extraordinary. Um, So if you layer into that pregnant woman, you're consuming fortified grains, you're taking your multi, you might be eating liver if you're so savvy. You're drinking these milks that might have extra folate and B12, and then you're also, using soy products. Um, you could overwhelm, you could absolutely imbalance the methyl donors in that delicate, delicate, delicate time.
Right.
So it's worth paying attention to. So this was going back to your question whether liver is safe in pregnancy. I would say absolutely it is, but you need to be paying attention to overall nutrients.
Right. I love that point. I love the point. And whilst we're kind of talking about, I guess we're kind of talking about, someone that is obviously in our instance is kind of, you've got a child in utero. What about, for instance, if I was to kind of think about how these kind of DNA expressions can happen, what about something like intense psychological experiences that, for instance, some people that, for instance, biological essentialist would say, that may have little impact. But how does something like psychological experiences, how does that impact DNA
Yeah.
expression? I'd love to know that.
Yeah, so there's a growing body of literature demonstrating in humans. I mean, we certainly have a pretty solid body of literature in animals, but a growing important body of literature in humans that psychic experience will absolutely influence gene expression in offspring, and not just that first generation offspring, but second and third subsequent generations.
Wow.
Famous studies include Holocaust, offspring of Holocaust survivors, and you can see a pattern of gene expression associated with sort of an imbalanced stress response, leading to, the chronic, so imbalanced stress response on genes one of the outcomes can be an increased rate of the chronic diseases of aging. So this imbalanced stress response can cause an inflammatory phenomena that increases the risk of heart disease and so forth. And you can see this in offspring of the Holocaust. You can see this in offspring of individuals from the World Trade Center bombings. Actually, that's been studied as well extraordinarily. Yeah, I
Wow.
know, I know.
Wow. Wow. Wow.
You can, there was a really interesting study that I wrote about in the book, actually two, um, Project Ice Storm. So this was in Quebec in Canada, for a 2-week time in the 90s. They had this massive ice storm. Everything was stopped. And it was very stressful. No power, and just the world shut down there in this very, very stressful moment of time in the 90s. Women who were pregnant gave birth to offspring who had higher incidence of asthma and autism, both. So gene expression was changed. And what's interesting is that they actually studied whether it was a psychological stress or a physical stress. And they showed that it didn't make a difference, actually. So it could have been the physical challenge of that environment, or it could have just been the psychic overwhelm of being in that. I mean, it's interesting. You know, a couple years ago, we lost power for a week here after a huge storm. We were fine. We were surviving. We had a little generator with a heater, but it was so stressful. It was so stressful. And it's just extraordinary to me that they're able to see that the type of stress really didn't matter in this population. Both were impactful. We weren't stuck outside. We didn't have the physical stress, right? We weren't going to die. We weren't going to freeze to death, but we certainly had the psychic stress of that experience, and it was potent. There's another interesting study called the Dutch hunger winter that's quite famous, and I wrote again about it in the book. And this is when Germany shut off food supply to the Netherlands during World War II, and women who were pregnant at that time gave birth to offspring, again, who had just the same kind of phenomena almost as you see with some of the Holocaust offspring and World Trade Center. So this increased risk of cardiovascular disease, of obesity, of diabetes, and so forth, with that early exposure. And that was a physical stress. That was starvation. That was really severe starvation. I think the average caloric intake during that time was something like 500 calories. So very low.
That's pretty remarkable. And you've given so many great tips today. For instance, we talked so much about, you kind of mentioned some of the things that we know day to day that make a difference: sleep,
Yes.
exercise, diet,
Yes.
meditation, Tai Chi, for instance,
Yeah.
and reducing stress. I'd love to pick up on stress if I can, because this is something that I've actually heard on this show. So kind of contrasting opinions about. And stress to me seems to be something that kind of this, I think that there can be kind of like a nuanced opinion on it. Clearly, as you said, chronic stress is a terrible, terrible thing. And it, as you mentioned, you talk about it in the book, it's a massive driver of aging. Some people have come on the show and they've said that short bursts of stress, acute stress, it can actually have immune
Yes.
enhancing effects. I kind of wonder if you kind of what your overall opinion of it would be, how perhaps how we should manage it, where it becomes dangerous. I'd love to get your take on that.
Yeah, in the best of all worlds, maybe I would have addressed sort of the hormetic benefit of stress, of short
Right.
bursts of stress. It isn't a toxin. It can be a resilience builder in those cases. I mean, we know the plants that are most rich in these all important epinutrients are plants that have grown in a stressful environment, like wild blueberries versus cultivated, and something called Himalayan tartary buckwheat, which is packed with epinutrients, and it literally grows in the sort of desolate conditions of the Himalaya, and survives, and it's packed with these really smart nutrients because the stress responses is what these compounds are being generated in abundance. Likewise for us, some stress experience will absolutely make us stronger. The stress of exercising, for instance. So I do touch on that in the book, and some other examples of hormesis. Yeah, the short-term stress of exercise, sure, it generates free radical activities, it generates more oxidative stress, but the cleanup that the body turns on after that experience is so potent, and so anti-aging. I mean, arguably, exercise is one of the most important longevity uh, activities that we can engage in. So
And what
yes,
type, if I could just ask there, what type would you say are the
so
best bang for a buck?
That's a really good question. If I were a weightlift, I am doing resistance training. I think both resistance and cardio are extremely important. And I think the literature is comparable on each. There's plenty of weightlifting scientists out there who will argue for resistance only, that's the most important thing. But cardiovascular health, the data on good cardiovascular fitness is very strong. So I think both are important. Where we need to be mindful is under exercising, not moving enough, or in some cases, there's some evidence that we can exercise too much. I cite one study in the book where these, power lifters had a pro-aging response to their event. They were competitive power lifters, and I was a competitive athlete in college. Um, and I can say, at the end of my season, I would always get sick. I mean, there's an immunosuppressive effect after you have a massive output like that. And if you look in the literature on that immunosuppressive effect and illness post competitive season, it's very well documented. It's very well documented. So I think that is an imbalance phenomena. I wouldn't trade it for the world. I loved it. It was important for me to do for so many other reasons. But it did have an immunosuppressive effect over the long haul. And I think in part, probably at that time, I wasn't training as smartly as I could have been. So under exercising is definitely a massive, massive problem. But in some cases, we need to be mindful if we're exercising, excessively.
And it's interesting you mentioned that because a number of academics I know, they tell me that throughout the university term they won't get sick, and then the summer comes, they think they can enjoy themselves, bam, they get struck down with some sort of illness. So
Yeah.
must
I think
must
that's stress. I think that's the influence of stress, and then the exertion of whatever activity that you're doing. Yeah, you can have an immunosuppressive sort of bang.
I love it. I think we've covered so much today. I mean,
Oh,
I've got,
let me give you just
please
one little
go on,
cool
please
pearl, because we're
go on.
talking about exercise. So exercise changes gene expression potently, as I've talked about, and it's a longevity agent for sure. If we're preconception, so if we're going to have kids, we're going to hand down some of that information, some of how exercise has influenced our epigenetics to our offspring. And I just think that's so cool. And other cool thing about exercise is that it almost acts like those methylation adaptogen nutrients, like the green tea or like the turmeric or rosmarinic acid and rosemary. It has these methylation adaptogen properties. It can turn on genes that were inhibited. It's exercise is extraordinary by so many different mechanisms related to gene expression and epigenetics.
Does anything else do that?
Does anything else pack
Yeah,
the
like, is there
get
kind
you?
of anything else that kind
That's
of
as
has that sense of,
extraordinary and
yeah,
I don't that's such a good question, and I would say off the top of my head, no. I mean, I'd really have to puzzle over that, but it's your vegetable. Exercise could be your vegetable. I mean, you can, there are inheritance patterns associated with it. It's really the most potent, probably, longevity intervention in some ways that we have. Or at least it's one of the best studied. I mean, I think nutrition is way up there, but um, I don't know that it's been studied quite as carefully as exercise. It's just, yeah, it's just smart.
I love it. I love it. And I would kind of love to finish with, I guess, kind of an empowering message, because I think your book is a really, really empowering one. And
Thank
I
you.
wonder, because the people listening to this, they might say, well, you know what, I've got a history perhaps of heart problems, or there might be some sort of disease within my family. Your book kind of says the opposite, that really we can take back control, that what we do day to day is
Yes.
essentially what matters most. So I wonder if you could kind of
Yes.
speak to that, about how we can essentially control so much of our health with day to day habitual things that we do every single day.
Yeah, 100%. Yes. We're in the driver's seat of gene expression. So epigenetics is the interface of how environment influences gene expression, and we are in the driver's seat of influencing what genes are on and what genes are off. And you're exactly right. It's our day-to-day choices and accumulated over time. Cool science is that sometimes one intervention, like they've studied exercise or meditation, and you can see just one time will have a favorable influence on gene expression, but it's that cumulative habit over time where you really make some profound changes. Um, but we're in the driver's seat. So if you want to take charge, know that if your parents or if your grandparents, have health histories that you're dreading or that you're terrified of, you're driving that car. You have so much say over whether you develop those or not. Yeah, it is extremely empowering.
I've absolutely loved this conversation. I got one question left for you that we sign off all of our podcasts with.
Uh-oh.
Uh-oh. Before I ask you to tell these guys where they can connect with you, all about the book, and anything else that you'd like to send our audience to, is the last question we sign off all of our interviews with is, what makes a life worth living?
That's a great question. What makes a so clearly, I would being connected to humans that you love in whatever that looks like, just being connected in a way that you love. It doesn't even have to be humans, actually. It can be your beloved pets, or so being well connected and following your passion. I would say those two things.
I love it. I love it. Where can these guys connect with you? Where would you like to send our audience?
Yeah, so you can come to youngeryouprogram.com. That's the website that will give you access into our worlds. And then drkarafitzgerald.com is my clinic practice, my podcast, all of our blogs, and it's just very information dense site, which will also bring you to Younger You and the Younger You site. So maybe Dr. Kara Fitzgerald is where to go.
Everything will be linked below for our audience listening. They can just swipe up on this episode. Everything linked today, discussed today, will be linked below. If you're watching on YouTube, then just click the description. Kara, this has been such a pleasure. There's been so much good, really actionable, mind-blowing science. There's been we've had it all today. So I can't thank you enough for just bringing so much value to our audience and for writing such a great book. I've loved reading
Thank
it and getting
you.
to know you
Thank
more.
you. Yeah, thanks so much for having me on. It's been a lot of fun to talk to you.